Ja. Blackbird singing in the dead of night. Take these broken wings and learn to fly. All your life, you were only waiting for this moment to rise. Blackbird singing in the dead of night. Take these sunken eyes and learn to see. All your life, you were only waiting for this moment to be free. Blackbird fly, blackbird fly, into the light of the dark at night. Blackbird singing in the dead of night. Take these broken wings and learn to fly. All your life, you were only waiting for this moment to rise. You were only waiting for this moment to rise. You were only waiting for this moment to rise.
Good afternoon, everybody. My name is Lewis Owen. I'm the Dean of the College of Sciences. Unfortunately, the head of Biological Sciences, Dr. Bruce Schwartzy, had an accident this morning and is unable to be here. He tells me it's something serious. I spoke to him about an hour ago, but I'm going to stand in for him right now. I was sort of scheduled to say a few things later in the afternoon. Firstly, I'd just like to welcome everybody in the room, and in particular welcome to any family, all friends, students, colleagues, everyone that's here. I'm incredibly honored to be here to celebrate the incredible life of Natalia Duché-Wilkins. Natalia joined our college as a faculty member in 2022. She joined the Department of Biological Sciences. She was an integral member of the department. When Natalia first arrived at NC State, we asked her to contribute to an article we were writing about new faculty. And here's what she wrote when she was asked why she chose to come to NC State for the next stage in her career. She wrote, "Every person I've had the opportunity to interact with, from students to staff and faculty, were kind, supportive, collaborative, open-minded, and passionate. To me, this reflects core institutional values that strongly align with my own." Reading this, it's obvious that Natalia had a profound love for NC State. She fitted perfectly into our community. But it also reveals a few core values and aspects of Natalia's personality. Those were acceptance, joy, and love, among many others. As you know, Natalia was a groundbreaking neuroscientist. She was dedicated to figuring out the interaction between the brain, the human immune system, and emotional well-being. This speaks volumes about her empathetic nature. She spent her career working to heal emotional pain. All of us will benefit from the work that she did for many years to come. Many of you are aware that Natalia's fiancé, Trevor has established Natalia Duché-Wilkins Memorial Endowment in her honor. This fund provides support for faculty, students, and postdocs in the Department of Biological Sciences. This support could include travel awards, stipends, enhancements, scholarships, and seminar attendance. Since this gift is an endowment, it will benefit biological sciences community in perpetuity. Natalia's legacy will live here in NC State forever. We are profoundly grateful to everybody who has contributed that so far. Trevor, we extend our deepest sympathies to you for your profound loss. And thanks will go forever to honoring Natalia with this endowment. So I want to thank you all. I also would like to thank today people here that helped to organize this event, particularly Gresham Fulner, Kurt Marsden, John Meiston, and Krista Baker. And also for our guest speakers that are coming here today, that have come here today, all the way from UC Davis, Brian Traynor, and from Michigan State University, Adam Moser and A.J. Robinson. So thank you all for coming here today. And I know that words cannot express our deep loss and the profound impact that Natalia had on our community in the time that she's with us. Thank you.
Thank you, Dean Owen. There are stars in the sky who are so far away that their light reaches us long after their initial fire has burned out. And even though that initial spark is gone, their light holds energy and their light holds power, even though the source is separated by expanses of time and space. And so it is with Dr. Natalia Duque Wilkins. She was a star, and she was a firecracker. And among many other roles, she served as a professor and as a scientist. And she gave her light broadly and generously. And that light that she gave you, whatever the form, whatever the medium, was her gift to you. It is yours to keep. And today, we celebrate that light that you hold at her memorial, her academic memorial symposium. An academic memorial symposium is a traditional gathering where we remember and honor one of our own, a scientist and a professor's contributions to research, service, and education. It's typically held months after passing so that we have journeyed through the initial phases of grief and we can concentrate more fully on that individual's impact. Today is made possible by the generous support of the Department of Biological Sciences. Thank you to Bruce Schulte, as well as the College of Sciences, and thank you, Dean Owen. And of course, we acknowledge all of those and thank all of those who have traveled long distances to be with us today. Okay, from California to Michigan and other places. We thank our student and staff volunteers who helped make this day possible. We thank the organizing committee, and beyond the organizing committee, we thank the administrators, staff, and faculty who have worked hard to make today a reality. We are grateful to all of you to help us with today. My heartfelt thanks goes to those individuals who have mentored Natalia's students, both in her research laboratory and in her classroom after her passing. I thank the team of students who successfully pushed for traffic calming on Clark Avenue. We have 3 stop signs. And I'm not so naive to think that that's going to solve all the problems on that, but it has slowed down traffic, and that is a win. And I thank those individuals inside and outside the university who have ensured her scientific legacy. And finally, I thank her friends, some of whom are with us today. I thank her family for sharing her with us, right? And we have individuals here with us today and also her Chilean family on live stream. And you are acknowledged. So typically a symposium features a series of talks, both formal and informal. Today will be mostly formal talks. There will be breaks in between the talks, as well as a reception and an after gathering. We ask that you direct your questions, if any, to the speakers during the breaks and the reception. And so today, over the next few hours, we are going to feature those individuals who can provide perspective on Natalia's journey to lead her to establish her own neuroimmunological laboratory here at NC State. The afternoon, in a few minutes, we'll begin with Dr. Brian Traynor, who served as Natalia's PhD advisor at the University of California, Davis. It was there that Natalia first studied the social and anxiety behaviors that captivated her. She worked on the underlying neuroendocrine mechanisms, and she worked on how adverse developmental environmental events, and in particular stress, negatively impacted later life outcomes. And then from there, we're going to turn to Doctors Adam Moser and A.J. Robeson, who advised Natalia during her postdoctoral studies at Michigan State University. And it was at Michigan State that she began to seriously incorporate the immune system into her research program, showing how adverse environmental effects early in development could induce, in her own words, long-term immune gene changes and heightened sensitivity to adult stress-induced behavioral and meningeal immune responses. And most importantly, that these effects could be mediated via mast cells. And you are going to hear the words mast cells over and over and over again today. Natalia was obsessed with mast cells. And when we talk about her as a scientist, we cannot separate out the mast cell from her work. Mast cells, they are the immune system's first responders. They're kind of like the alarm system. They're located throughout the body. They're in the peripheral areas like the mesentery, the skin, the spleen. And importantly for behavior, for the neuroscience side of things, they're also located in the brain and in the meninges, right, which helps to interact with the brain. And so when they're activated, mast cells release chemicals like histamine. They can fight infections. They can induce allergies. They can promote wound healing. They can regulate inflammation. They do all kinds of stuff, people. And most importantly for Natalia, they could influence behavior. And so when she first came to NC State, and I remember her symposium, I remember her talks that she gave, and I remember when she was fighting for her laboratory, she pushed hard for an integrative creative research program based upon the premise, and again, her own words, that mast cells represent a vulnerable fulcrum by which developmental exposure to an environmental contaminant impacts adult social and anxiety behavior. And she was grounded in this premise, and it drove her hard, okay? Her goal was to make an impact. She wanted that. And yes, part of that impact was to connect the environmental insults to the immune cells, the mast cells, and neural systems to behavior. But really what she wanted was to make a difference in her teaching, research, and service programs that she could be proud of no matter what. And she had conversations about that with person after person after person at NC State. And there is much to be proud of that she accomplished. By the time of her death, her drive led her to the central hypothesis, and I believe that Heather Patterson will talk about this a bit later, that developmental exposure to environmental insults, and in particular at that point in her development, she was working on a flame retardant named Firemaster 550, that these developmental exposures, these insults, reprogram some mast cells into a hyperactive state, that they are turned on, leading to tissue-specific, in her own words, functional alterations in mast cells within the brain and other tissues. And she argued in her first independent papers, which she achieved before her death, and in a fundable scored grant application, which she also achieved before she passed, that this mast cell dysregulation amplifies the neuroinflammatory responses to everyday immune challenges, ultimately increasing the susceptibility to those neurodevelopmental disorders with impacts later in life. Her findings highlighted mast cell hyperactivity as a key novel mechanism potentially connecting environmental exposures to the growing prevalence of neurodevelopmental disorders. She was helping make that connection. And we have many local speakers. Heather, I've already mentioned, we have her students, we have faculty members from NC State who are going to provide perspective on this phase of her journey and help us understand how she got and the impact that she made, as well as words from Trevor. So afterwards, we'll enjoy a light reception. And for those who choose, the after gathering will be at the Raleigh Brewing Company around 8 p.m. Okay. So now it is with great appreciation that I introduce Dr. Brian Traynor, professor and director of the Behavioral Neuroendocrinology Laboratory at UC Davis, and he was Natalia's PhD advisor. He earned his bachelor's from UT Austin, his MS from the University of Nebraska at Lincoln, his PhD from the University of Wisconsin. And Brian, please come on up. Brian is the leading expert. Brian is the leading expert in the underlying neural mechanisms of social behaviors that are related to mental illness, especially from an ethological perspective that incorporates variables like sex and stress. Brian Traynor.
Thanks, John. It's good to be with you all today. I'm going to tell you a little bit about Natalia's discoveries from her graduate training and how she touched a lot of our lives there. So I first met Natalia in 2014, and she had been at UC Davis for 2 years, and she was actually in a different lab, and it wasn't really going very well. And so the graduate advisor of her program set up a meeting between us, and she told me she wanted to switch into my lab. And I already had 4 graduate students, and my first response was, well, why don't you just sort of stick it out a little bit longer? Maybe things will get better. And sometimes I think about that moment. It's sort of like someone walks into your office here, this 5-star recruit, and says they want to work with you, and you say, why don't you go over to UNC? So lucky for me, Natalia said, no way, I'm done with that lab, and that's how it started. So she joined at a pretty opportune time. I did have 4 grad students at the time, but 3 of them were going to be graduating. And Michael Steinman, he was my very first grad student. He needed back surgery, and he couldn't finish the last experiment. And so Natalia worked with, you can see her there second, from the left over there. She worked with Rebbe Howe, who's fourth, 2 people down from Natalia, to do the last experiment looking at oxytocin. So I'm sure most of you have heard of oxytocin before, and what usually comes to mind is sort of affiliative behaviors, sort of more positive social contexts. So parent-offspring bonding. These are drawings that Natalia made. I'm going to use a lot of her drawings today. And a major question in the field, and then also this is pair bonding over here. And so a major question in the field, though, has been to explain why sometimes oxytocin can promote other types of behaviors that we might call sort of antisocial, so social avoidance, or a lack of trust. These are studies pretty well known, replicated in humans. And so they tend to get ignored sometimes because how can one hormone promote sort of affiliative social interactions, but also have these very different behavioral effects? And so a major advance in the field was this idea called the social salience hypothesis, which basically says that oxytocin makes good experiences better and bad experiences worse. And so several groups had floated this idea, but no one really knew how it could work in the brain. And so one of the main methods we use in my lab is we study social stress. And social stress can happen when an individual loses an aggressive interaction. And we study this because when individuals lose an aggressive interaction, they avoid new social situations. And so you can see this in mice and rats, fish, birds, even primates. And so social avoidance is an important behavior to study because sort of a withdrawal from social situations is a symptom that's often associated with anxiety and depression. Now, Natalia helped us finish a study that showed that social stress increases the activity of oxytocin neurons in certain parts of the brain. And this paper was accepted into a journal called Biological Psychiatry. And so if you haven't heard of that journal before, it's pretty hard to publish in. My lab had submitted 2 papers to this journal, and both of them had been rejected. And so when we got this paper in, it was a super big deal for us. And so Natalia volunteered to draw this cover art that you see here. And I think it looks like you're going to hear a lot more about her artistic capabilities. And so this drawing is more than just like a nice drawing. I'm going to tell you that scientifically there are concepts in there. So you can see this one mouse at the bottom is avoiding the other two. But it's not just avoiding it. See how it's sort of oriented towards it. It's looking at it. And so today we call this behavior social vigilance. Now, Natalia drew this after being in the lab barely 2 years. But when she made this drawing, we weren't like thinking about social vigilance as a thing we could measure. In fact, it was only one of Natalia's later papers where we actually sort of figured out that we could measure social vigilance, and it's something that we routinely measure in the lab now. And this is a behavior that we think is related to anxiety behaviors, because children who have behavioral inhibition or sort of anxious temperament, children do this too. And behavioral inhibition is the #1 predictor of the development of an anxiety disorder later in life. And so I feel like this image here, I love looking at it, one, because it represents a big victory for the lab, but also because it makes me think about Natalia's intuition and how she could see things that the rest of us couldn't see sometimes. Now, she built on this intuition in her dissertation, where she made really important discoveries. And I think her paper, we published again in Biological Psychiatry with her paper in 2018. I think this is probably the most important paper my lab has put out. And the reason is she discovered like a totally different way of reducing anxiety-related behaviors. Let's see if I can use my mouse here. So Natalia used the stress paradigm to show that, I'll just gonna, oh, here we go, all right. Here we go. All right, so I haven't used a pointer in ages. I usually teach, my main teaching class is 400 people now, and so it's in this giant thing, so I can't use the laser pointer. I'm gonna try and remember. Anyway, so in the blue bar, you can see stress is reducing social approach. This is something we had found a bunch of times. Now remember, in the previous study I told you about, oxytocin neurons were more active after stress. And so what she showed is she could give a drug that blocks oxytocin receptors to these stressed animals, and it could reverse the effects of stress. And that's what you see here in this red bar. So she gave a single injection of this drug that blocks oxytocin receptors, and it's like those mice had never been stressed. Now, you can get this effect by using an antidepressant like fluoxetine, Prozac, but to get that effect, you have to treat those mice every single day for 4 weeks, just like in humans, right? And instead here, she blocked the oxytocin receptors once and acutely achieved the same effect. It's a groundbreaking discovery, and no one would ever think to have used, would have thought, everyone usually thinks oxytocin promotes social interactions, and here she's finding that you can block them and it can have a sort of an anxiety effect. And so anyway, this is a finding that we're trying to follow up on. The drug that she used has actually passed phase 1 clinical trials in humans. So in theory, if some of our next round of experiments in mice were successful, we could then test this out in humans. Now, this by itself, I think, is a really important result, but in the same paper, Natalia also showed in the brain how it could be working. And so when she was in grad school, the field was thinking that oxytocin could increase salience, social salience, by acting in the dopamine system, which was a good idea, because the dopamine system can be activated in both positive and negative social contexts. But Natalia showed that this idea wasn't exactly right. And this is a diagram that I use in all of my talks, and maybe you can recognize more drawings that Natalia made. And it shows how oxytocin does act in the dopamine system to promote sort of affiliative social approach behaviors. But it also shows what she discovered, which is that oxytocin can act in the extended amygdala to drive social avoidance behaviors. And so when you ask the question, how can oxytocin have such different behavioral effects? How could it promote social approach or social anxiety, right? The answer that Natalia helped put together is that it's acting in different parts of the brain. And she also showed that the oxytocin that's acting in the extended amygdala comes from a different source. It comes from outside of the hypothalamus, where most oxytocin is made. And she used a tool called morpholinos to do this. And so what a morpholino is it's a strand of DNA that matches up with the oxytocin mRNA, the gene expression, and it prevents the oxytocin mRNA from making the protein. So what you can do is selectively, in a specific part of the brain, reduce oxytocin production. And she used this in the extended amygdala to show that oxytocin made outside of the hypothalamus could actually drive social avoidance after stress. And so even though many people had seen these oxytocin neurons outside of the hypothalamus, no one knew what they did, and she was the first one to show that they actually did have a behavioral function. And to do this experiment was really hard. We had never used these morpholinos. The first time she tried copying a set of methods that was published in a paper, and it didn't work, and she had to come up with her own protocol that did work. And then she was also the first person to use viruses in my lab, and so she used an approach where she used a virus that would make this green fluorescent protein only in oxytocin neurons, and so she put that into the extended amygdala, and she could see how they were connected in the brain. And so you could see these little threads of green fluorescent protein here, and this shows you that this part of the brain is connected to those oxytocin neurons outside of the hypothalamus. Now, this series of papers, these are seminal contributions to the literature. People know about me and my lab because of these papers, but that only really scratches the surface of who Natalia was as a scientist. Because part of what gave her unique perspectives in science was her training as a veterinarian. After Natalia had been in the lab for a few years, she came to me and told me that she thought the cages of our mice were too small. Now, the mice that we study in my lab are quite a bit bigger than the standard sort of laboratory mice. And I think in my heart, when she told me this, I think I knew she was right. But I'm running the lab, and my first response is, well, what if it messes up our science? Now, probably those of you who know Natalia in a science environment, that she's not very good at taking no for an answer. And so she successfully applied for a grant for $5,000 from the American Association for Laboratory Science. And then using that money, she got bigger cages with extra toys for them. And then she ran an experiment where mice were randomly assigned to the cages, and then she characterized developmentally as they grew up, from when they were weaned all the way until they were adults, and showed that the bigger cages actually did improve their welfare. They did less of this sort of repetitive backflipping behavior. And then not only that, she showed that not only did the bigger cages improve the welfare for the mice, but it actually made the science better. We would actually get cleaner, stronger results in the stress studies. So even though I didn't want to make the change because I was sort of like, oh, what if this messes things up? Because she went and got that grant, she showed that the science was better. And so after she showed me those data, I spent thousands of dollars to put all of our mice in the bigger cages. And whenever I go into the mouse rooms and I see those cages, I think of her, because she made that happen. And so that's part of her legacy as well in Davis, not just in the science and the individual lives that she touched, but all of our mice there actually have a better life because of her. Now, this study was actually run by an undergraduate, and so that touches on how she was as a mentor. This here was Natalia's dream team. Sae Yokoyama did neurobiology experiments. Vanessa Minnie ran those enrichment studies that I just told you about. And Mary Pham did a lot of the behavior with social vigilance in her big 2018 paper. And all three of them made huge, tangible contributions as undergraduate researchers. But you didn't have to have the big home run in the lab to have a good mentoring experience with Natalia. A lot of her students, maybe they didn't work on a big experiment, but many of them were very successful in other areas. Her mentees went on to have careers as mental health professionals, physicians, and also in technology. And even after she left the lab, she was a very generous colleague. During the pandemic, I was hosting a student from the Netherlands, and just as that student was getting good at surgeries, the lockdowns came, and so she couldn't work on any experiments. And the student wanted to write a paper on social buffering, and I kind of talked her into doing some social contagion research. These are not like main areas of focus for me. And we had this data set that Natalia had sort of created on the side, kind of combining data from different studies, and there was an interesting result there, and we wanted to use that data in the paper. So I asked Natalia, would she want to be involved? And she said yes. And so we sent her the first draft of the paper. And so she was excited, but here is what she said. I went over the manuscript, and I have a lot of comments. Sorry, I feel all the information is there. It just needs to be reorganized to make it clear there's an underlying hypothesis. I'm missing a clear direction in the writing and to make it easier to read. And she kind of tore it up. And this went on for about 12 pages. So I don't know if any of the students here experienced this with Natalia, but if you did, you're not the only one. And so when it was done, the paper was way better than when it started, and it sailed through review at Hormones and Behavior. And Natalia drew these beautiful rats for one of our figures, and the editor chose it for the cover of the journal. And so what started as a side project kind of ended up actually being, over time, has become a strong contribution to the literature. This is a graph that shows how many times the paper's been cited since it came out. Hormones and Behavior isn't one of those high-impact journals. It's like a solid, society-level journal, but people are reading this paper and citing it. And I was at a conference once at Ohio State. Actually, I think some of the grad students were there. Is where I met them, and I saw that figure on somebody's laptop. And I was like, whoa, that's cool. Well, it turns out it was this man. It's Rui Oliveira, who is actually a well-known professor in Portugal and a leader in that field of sort of social contagion. And people in my field know about Natalia for those big papers that I was telling you about on oxytocin, but getting those papers was not what drove her. It was sort of a curiosity and a love of discovery. And I feel like this paper, while not like central to my research program, is a great example of sort of the contribution that Natalia could bring to science. And so back in Davis, when we would discuss her career path, Natalia would tell me, Brian, I just want to do science. And I was so happy that she got the position here. And it was even better to know that all of you were seeing the Natalia that I knew. And she sent me this performance review a couple months ago. And it describes her as integrative, brilliant, tenacious, and talented. And the primary advice was just keep doing it. I just want to thank John and everyone that was involved in writing that report. Being an assistant professor is hard. There's a lot of struggle that comes with starting a new lab, a lot of self-doubt, rejection. It's all, those of us that have been faculty members, we've all been through it. And, sometimes you can doubt yourself a little bit. And just reading this brought tears to my eyes. And I know it made Natalia feel great about herself, too, and the direction her lab was going. And, if it's not obvious by now, we texted a lot. And I know she was happy here. Like when she sent me this text here about the lab party where the students made fun of her. I loved it, that's what she said. All right, wants to know if I want to change my password. All right, let's see if I can. All right, I'm like one of these professors that, all right, there we go. All right, so it's hard to know how to move forward when you lose someone so special. And so I'm trying to lean into the things that she taught me. When an undergrad asked me about testing a new method for improving the housing of the mice, I said, let's try it. I'm trying to take more risks in the lab, and I'm just trying to make the most of the time I have now. And in Davis, we planted this tree for her in North Star Park. And if you're ever in Davis, look me up and we can go sit there. Every time I've been there, there's bluebirds flying around or turtles. It's a really peaceful place. And a few weeks ago, we got together to paint some rocks to place around the tree. They turned out to be really beautiful. And the day that we did it, it was a rainy day. We had to squeeze into the garage. And there were people there who were Natalia's close friends and mentees. and people who only met her once at a conference or a Zoom meeting, and some people that only knew about her from papers or stories that I would tell. And for me, looking at these pictures get at the essence of who Natalia was, which I think bringing people together to create something amazing. Thank you.
Thank you, Brian. We will take about a 10-minute break, and then we will return with our Michigan State speakers. Thank you. 10-minute break.
It is my pleasure to introduce both of Natalia's postdoc advisors. I'll start with Dr. Adam Moser. So Dr. Moser is with us today from Michigan State. He was one of Natalia's postdoctoral advisors. He received his bachelor's from the University of Massachusetts Amherst, and then his master's in animal science, his PhD in physiology, and his DVM all right here at the vet school at NC State, where he was also an assistant professor of swine health and production. So he was with us for many years here, and then he moved to Michigan State, where he is now the Matilda R. Wilson Endowed Chair in Large Animal Clinical Sciences and the director of the Gastrointestinal Stress Biology Lab. His lab studies brain-gut communication and neuroimmune interactions with an emphasis on mast cells.
Okay, well, thank you for the introduction. Honored to be here today. And I'd like to reflect on Natalia's work, the impact of it, and her legacy as part of my portion of the talk before AJ will take over. But I think to really understand her legacy with us, this part of her legacy, I'm going to tell you about how AJ and I began our collaboration, because that's really where Natalia came into our lives and really had that impact with us. So just to tell you about this, at that time, my lab was studying early life adversity and gut disorders in life, and I had this particular interest in passion in mast cells. And we talked, there's the name, mast cells. We're going to start hearing this again, and I was so honored to hear, that Natalia was so excited about these mast cells, because they're these really cool cells, and I'm just honored that she really got into those cells and really made her own research program out of them. But I was studying that, and AJ, we started talking, and he's studying neural circuits of stress associated behavioral disorders, and we decided we really need to collaborate, because there's a lot of things that we're studying that are kind of similar, and there's going to be a lot of synergy in this project and what we're talking about. And so after we decided we're going to open up a joint postdoc position in our laboratory. So we were thinking about a scientist that can work equally across our two labs and bridge this gap and really drive a vision forward. So what we were looking for is an individual who could actually span that sort of gap and be willing to take this leap in this kind of untypical environment, and to work seamlessly integrated in between two labs to learn two new fields of study, and learn new methods, and at the same time help drive this one mission between our groups. And when we put out this ad, I think AJ and I were talking about it, we were like, we're never going to find someone like this. This is such a rare quality that there's nobody out there. And then there's Natalia. We talked to Brian Trainer and about her, and she ended up being the perfect fit for that position. And there was nobody else like her. She took this position. She had the courage to just take that leap into the unknown she saw this opportunity, and just her qualities of her, ability to, just her curiosity or genuine curiosity about science, her tenacity and passion about science, her ability to work with all types of people in different programs, her just thirst for learning. It really took something unique and she made it work, and she ended up driving and building this bridge in our program, which was absolutely amazing. And for me, for my lab, I was studying mast cells, and she took to mast cells very quickly, and she learned this field, learned the techniques, became very proficient at this, and through our collaborations with AJ's lab, really set forth a foundational paper in studying the role that mast cells play in early life adversity and associated neurobehavioral disorders and outcomes such as anxiety and depression. Because before that time, before her paper in this area, it was just really a fragmented field. Mast cells were activated. We knew that early life adversity was linked with all these diseases, but her work was the first to really make that functional connection and show that mast cells were these critical immune cells that are playing an important role in depression and anxiety. And one of her papers showed that when you treat with mast cell stabilizers, you could reduce some of these neurobehavioral and anxiety outcomes. So this was a pretty big foundational study, and it was the first of its kind. And this set a foundation that we're still doing work from. This is something that was solidified, and we're still doing work in this area. And AJ is going to talk about some of her other major impactful studies that she did in our laboratory. And and she's also began to develop these models of looking at the role that mast cells play in general, just in behavioral changes in mice. And this is pretty novel. Again, this is something that hasn't been functionally shown before, and she has laid the groundwork. And I was so impressed to see how the growth of her program when she came to NC State, and it was just this past year when she visited us at MSU. We all had dinner with her, and she gave a talk, and it was just I was just so happy to see how she became her own scientist, and she took what she learned from her previous mentors and really built her own career. And yeah, it was just amazing to see. And I was also, as a personal note, I was happy that she came to NC State. As was mentioned, I have a personal connection to NC State, and I knew that this would be a place that she could thrive, and her program would do well. So I was happy to see that she came here, and she was going to be in a good place. So when I think about Natalia, I think about her legacy that she leaves, because it's not so much through the papers or the data that she generated. It was through her connections and building bridges across people and laboratories, and her training of her students, training the next generation of scientists, her inspiring questions that were collaborations were started amongst faculty and everybody that she worked with. And that's one thing that if I had to take one thing from her life, it's this. It's just having the courage to really make a significant impact. It's to have the courage to go into areas that are uncomfortable, to challenge boundaries, to create connections between people and groups, to build things that are new when originally they were just seen as obstacles or no connections at all. And that's what Natalia leaves with me, and I think leaves us with all. Thank you.
Next, I'd like to welcome Dr. A.J. Robeson. A.J. Robeson received his bachelor's in biology from Rhodes College and his PhD from Vanderbilt. He then did postdocs at Vanderbilt, UT Southwestern, and then Mount Sinai School of Medicine. Since 2013, he has led his lab at Michigan State University, focused on understanding how drug addiction and chronic stress influence gene expression in the brain. He is the director of the neuroscience. He's also the director of the neuroscience program at Michigan State.
Hi, everybody. I thought I would start a little bit by talking a little bit about Natalia's science, and then maybe talk a little bit about her as a person and what she meant to my laboratory and to me. So to tell you a little bit about Natalia's science, I just wanted to focus on the project that she led in my laboratory in collaboration with Adam's laboratory that really advanced our understanding of how mast cells work. And my laboratory studies gene expression changes, and up until I met Adam, my lab was studying gene expression changes in neurons and discrete circuits in the brain. But when I started working with Adam, I started thinking about how gene expression, genes being turned on or off, how that regulates the activity of mast cells as well. And Natalia really led this project in my laboratory. Everybody's mentioned mast cells. This is the picture that Natalia always used to describe them. It's the big purple cell there. It's a gorgeous cell. When you were in 6th grade and your biology teacher explained what a cell looks like, they told you that it had a big nucleus in the middle, and then it had these mitochondria, the powerhouse of the cell, and it had an endoplasmic, and you learned all these organelles and stuff. Mast cell doesn't look anything like that. It's got a big nucleus in the middle, but then it's got all these things that we call vesicles. They're these kind of packages inside it, like balloons. And these balloons are filled with all kinds of stuff. Some of them are filled with enzymes, like proteases and tryptases and chimases, and these things, when the mast cell releases them, they break down connective tissue. They destroy other objects or cells. They regulate the function of other cells. It has neurotransmitters in it, like a neuron. Things like histamine and serotonin that it uses to communicate with other cells, including neurons, as Natalia was beginning to uncover. And then it has all kinds of immune signals that it uses to orchestrate immune responses. Because it has all of these different capabilities, because it's so multifaceted in all of the different things it can do, and I am not an expert in this, everything I'm saying right now I learned from Adam. Because it can do all of these amazing things, it orchestrates the function of all kinds of different things in your body, but also in the bodies of mice, where my lab studies it, or in the bodies of pigs, where Adam's lab studies it. And what these mast cells can do in the periphery of the body, meaning all out here, it can regulate how you respond to bacterial infection. Or what it's really famous for, what mast cells are really famous for, is regulating how you respond to allergens, like the pollen that you're going to start smelling and reacting to all the time in the next couple of months, especially in places like the Arboretum. The mast cells are what regulate the runny nose that you get and the swelling and all of those terrible things. So sometimes we hate mast cells. But they also help you respond to bacterial infections and other infections by regulating how the immune system works. What Natalia was trying to figure out is how does in my lab and Adam's lab, is how does repeated activation of these mast cells, like what happens when you get an infection or when you are exposed to allergens, how does that affect their sustained function? And is that something we can reach inside the body and control to regulate your response to allergens or to infection? Can we develop methods that will let us reach inside the mouse's body, and then maybe eventually human bodies, to control allergic responses in ways that would be breakthroughs for people with terrible allergies or people with mastocytosis or other diseases related to mast cells. That was Natalia's goal in this project and Adam and I's goal in this project. And I just want to very briefly just show you a couple of pieces of data about how she was figuring out how we could maybe do that. I study a gene called FOSB. I've been studying this gene since I started my postdoc with Eric Nestler back in 2008. And one night I was at a bar with some friends, as I am wont to do, and there was this tall, kind of goofy guy at the bar who was really into sports, and we started talking, and he introduced himself as Adam Moser. And he said to me, "I study mast cells," and I said, "What's a mast cell?" He explained everything to me that I just explained to you about them. And he said, "I'm trying to figure out what genes are turned on and off in mast cells when they get activated." And I said, "How are you doing that?" He said, "I'm doing RNA sequencing and all of these kinds of really cool, brand new techniques that nobody else had ever applied to mast cells before." I said, "Oh, that's interesting. What genes are you finding?" He said, "Obscure, weird genes that nobody's ever heard of." I'm like, "Well, what's the #1 hit you've got?" And he said, "Well, it's this thing called FOSB." And I about pooped myself right there at the bar. There's 22,000 or so genes that a mouse makes, and the one that he was studying was the one I'd been studying for 15 years. And I said, "I've got all these tools. I've got knockout mice, I got flox mice, I got antibodies, let's do this." And so we started that project that Adam kind of described to you. And as he said, we needed somebody to drive it forward because we're lazy and we're not that smart and we weren't going to do it ourselves. We needed somebody who could come in and do this work in our labs for us, and that's where Natalia came in. And Adam described how that worked, how we were so lucky to know Brian Traynor and to have him give Natalia a blessing that our labs would be a good place for her. And she took his advice and came and joined us, and this is what she found. What she found was that when these mast cells become active, as Adam had described in his RNA-seq studies, she confirmed, along with some other folks in our groups, that indeed the mast cells would make more of this delta FOSB protein, right? And when they did this, it would have some effect on the function of the mast cells. And the way she tried to figure out what that function was is that she took the FOSB away from the mast cells. She mutated this gene, but only in the mast cells. This gene's made in neurons. That's where I'd been studying it. She didn't mess with it in the neurons. It's made in some other cells too, other kinds of immune cells. She didn't mess with it there. She exclusively prevented the expression of FOSB in mast cells. And when she did that, she found that these mast cells were hyperactive. See, they're releasing more histamine. That's the stuff that makes your skin get red and your nose run when you have allergies. That's why you take an antihistamine to block your allergies. She found that when she took away the FOSB gene, these mast cells were making way more histamine, or releasing way more histamine. And not only when she had them in the little petri dishes, but when she did this in the living mouse, when she took the FOSB gene away from the mast cells in a living mouse, that mouse had more histamine in its bloodstream, right? She also found that these other immune signals that the mast cell gives to help fight off infection, those are also higher in these mice where she took away the FOSB from these mast cells. And she could see that here as well. When she gave the mice an infection, it's not a real infection. She's tricking them. She's giving them a bacterial protein that makes the mouse's body think it's infected. She has a much bigger mast cell response when she took away this FOSB gene. So then she took these mast cells and she looked at them using a technique called electron microscopy in collaboration with some other friends at MSU. And she saw that in the regular mast cells, they have all these little balloons in them, these vesicles I told you about, and the vesicles are filled with all this stuff, the histamines and the prostaglandins and the enzymes, the chimases, the tryptases. You can see them all in those little vesicles. When she looked at the ones where she had taken the FOSB away, look at the vesicles. They're empty. It's because they've been dumping all of this stuff out. These mast cells are hyperactive when she took this gene away, when she stopped this gene from functioning. You can see how the mast cells make this FOSB gene. This is the protein that they're making. Here's where she knocked it out, right? So these are the mast cells that aren't making that protein. When she looked at these mice, their body weight is normal. They run around just like other mice do. But if she gave them the infection, right, she tricked them with that bacterial protein, they had a huge drop in body temperature, much more than a normal mouse would, right? And that's because they're having a much stronger immune response because their mast cells are hyperactive. When she looked at how sick they were, this is the mouse's clinical score, they had a much stronger sickness in response to this bacterial infection. So what she discovered, essentially, and there's a bunch of other data that I'm not going to show you, is that the mast cells, when they're activated, they make this FOSB gene. And what this FOSB gene is doing is it's suppressing their function. It's making them not as active anymore. It's preventing them from having runaway activity. And when she took the FOSB gene away, that's exactly what happened. They had runaway activity. So the idea then was, can we functionally control this FOSB gene, or are there things this FOSB gene is doing downstream that we could functionally control to act as new treatments for things like allergy and infection? And so, again, I don't have time to show you too much, but what she did next was to try and figure out which genes the FOSB was controlling, what was happening in these mast cells transcriptionally to change their activity. And this volcano plot, if you've ever seen one of these before, shows a whole bunch of genes that are upregulated and a couple that are downregulated as a result of FOSB being missing. And she combined that with another assay that showed which genes FOSB was binding to and directly controlling. And she was able to pull out a few targets. These are a couple of them. These are a couple of different genes that she then went on to study. And one of them turned out it wasn't a real target. She did her validation experiments, looked the same in the knockouts and the controls. But the other one, this gene called DUSP4, she found that in a regular mast cell, that it gets highly upregulated when the mast cell's active, but when she took away the FOSB, it wasn't activated anymore. It's very clearly a downstream target of this FOSB gene. And she then went on to show that this DUSP4 is important for mast cell function. And there are people in my lab right now, Brianna Smith, a student, and Emmanuel Kwanzaa, a postdoc, who are working on figuring out how this DUSP gene that FOSB was regulating is changing the function of mast cells and whether we can actually control that. So the legacy of this in my lab from a science perspective is that we're trying to figure out these downstream genes and whether we can reach in and control those genes as a way of combating allergy or infectious responses. And then we're also trying to drug this FOSB. We have a 15-year grant now that's trying to create compounds that bind to this FOSB and prevent it from doing its function or cause it to function even better. Now, if you've been following what I've been trying to show you about Natalia's work, you'd think to yourself, if we made a compound that prevented FOSB from working, that'd be bad for mast cells. They'd be hyperactive. They'd work too much. And so that's not what we're looking for. What we've found over the last year is 3 different drugs that bind to FOSB and make it bind to DNA even better and make it regulate downstream gene expression even better. It's my hope that some of those drugs, as we refine them and make them better and test them in cells and mice, will eventually get to the clinic and potentially be drugs that can suppress mast cell activity in a way that is different from the way antihistamines prevent the downstream function of mast cells. And I think it's a possibility that in the next 20 years that kind of thing could happen, and that would stem directly from Natalia's contributions. So that's the science I wanted to tell you about. That science was, is a huge collaboration between myself and Adam Moser. And then there are a lot of people who contributed to that from my lab and Adam's lab, and we're funded by a number of different sources. But all of it feeds down into this name right here, Natalia, and she was the centerpiece for all of that work. I don't have that many pictures of Natalia, and it bothers me when I went and tried to look for a lot of pictures of her. This is an early picture when she had kind of first joined my lab and Adam's lab. You can see how much better looking we were. And then there's Natalia in the middle with some of the folks who were in our lab at that time at a lunch. And here she is the week before she passed away. She came to MSU and visited us, as Adam mentioned, and we had dinner together, and she stayed at my house with Michelle and I and our son Max, and we have 2 cats. And one of those cats is a gift from Adam and his wife, who breed cats because they're veterinarians. And that cat is really sweet and really fun, and we really love him, but he doesn't sleep with us. He always sleeps by himself. And Natalia came and stayed with us, and she stayed in our guest room, and that cat went in there and slept in her bed all night long. And that was not a surprise to me, and I don't think it's a surprise to Trevor to hear it either, or everybody who knew her. Everybody remember Snow White? Or maybe it's Cinderella I'm thinking of, but one of those Disney princesses from the 40s or 50s, and she's walking through the woods, and all the birds just land on her shoulders, and the deer come up and smell her, and the rabbits. That's Natalia. Animals knew, the same way that all of us do, that this was a person you just loved, and this is a person who's going to love you back. Here she is with Ken Moon, who's here today. He came down here from Michigan. Ken is the lab manager for my laboratory for the last 13 years, and one of Natalia's very close friends. And then here she is playing this dice game in her house with Trevor and me and Ken, and drinking a drink called Casacha. Am I right, Trevor? No, we're not far off. There you go, thank you. I won't try to say it. It's a liqueur that we were drinking that night. This was maybe 4 years ago, and I'm still a little hungover. And we were playing this dice game that involves lying to each other. And I think we probably played it about 10 or 12 times that night, and I didn't win one hand, and Ken didn't win any, and Natalia didn't win any. Trevor, I think, went 12-0 that night. And if that tells you a little something about Trevor's personality, I hope that you'll learn a little lesson from that as well. All right, enough jokes and stuff. I'm not going to cry, but I'm going to get close, I think. Natalia, I'll talk about Trevor one more time in saying that, Trevor was a professional athlete, so he'll know this term that I'm about to use, which is glue guy. When you have a team, you have people who might be really good scorers in basketball, and others who might be really good defenders, or somebody who is a great blocker in football, or somebody else who's a great runner. But you also need people on that team who make the runner and the blocker work together, and the shooter and the defender work together. Even if they don't like each other, you need a person who's going to make them function together well, who's going to bring them all together. And in sports, oftentimes we call that a glue guy. Natalia was a glue guy. In my lab, and Adam's lab, she brought everybody together. And she did it scientifically because she thought about every possible scientific concept she could and was fascinated by every scientific thing she ever saw. And so she brought everyone together because she had a common interest in everything everyone was doing. But she also brought everyone together because of her incredible passion. She was one of those people, what we struggle with a lot of times as mentors is, when you have a graduate student or a postdoc or a technician who sees this work as a job, and they're just trying to get it done and get out of there. Or they're not really that interested, but they know they got to do this to get through to their degree or to get their paycheck or whatever it is. That happens sometimes. But then you get one like Natalia, who you can't keep out of the lab. You couldn't pry her out of the lab with a crowbar. She was there late at night, she was there on the weekends, and she wasn't there just because she wanted to be super successful, which she did. She was ambitious. She was there because she loved being there. She loved the science. And Brian says it all the time. Brian is a very close friend, and we bonded over many things, but I think Natalia brought us together more than anything else. Brian said all the time that Natalia would tell him, "I just want to do science." She lived that way. She really did just want to do science. She wanted to do it all the time. The word I use all the time for her is florid. It's kind of ironic to use that word in an arboretum. I think what it really means is growing a lot of buds and flowers and leaves, but it's metaphorical, I think, in this sense. That florid means she had a million ideas. There were things growing out of her brain all the time. I felt like my job and Adam's job and Brian's job as her mentors were to help put the blinders on her sometimes and help give her some direction, because she could win a Nobel Prize in 10 different fields and was thinking about important science and exciting questions in a million areas all the time. And so our only job was to try and help her direct that incredible passion and that incredible florid brain of hers to be productive. And I think, you know, to what little bit of contribution we made, we were successful in that way in harnessing that incredible engine that she had and helping direct her toward the things that we've shown you today that she contributed scientifically. The last thing I'll say is that I loved Natalia. She was a very good friend, but she was more like family. And being around her made me a better scientist and a better person. And I think that's probably true of everybody who met her. And I'm seeing a lot of nods when I say that, and I think that makes a lot of sense. All right, those are my thoughts about her. Adam, did you want to come back up and say anything else about how she brought our labs together? Are we kind of, did we cover all the bases? All right, I think before I tear up too much, I'll just say one more last thank you to everybody at NC State who's put this event together. A big thank you to Brian and Adam for you know, being partners with me in mentoring Natalia. And then the biggest thank you to Trevor, who also has become a very close friend, and for doing the work he's done over the last few months to cement Natalia's legacy, but also for bringing the joy to her life that he brought. And as a friend of Natalia, as somebody who loved Natalia, it made me very happy to see them together and to see what Trevor did in her life and the positive change he made in her life. All right, thank you guys very much for listening, and I really appreciate it, and I'll be thinking of all of you.
Thank you so much, AJ and Adam. We will now take a break for about 10 minutes. And if you have any questions about mast cells, now's your chance.
Okay, good afternoon. My name is Kurt Marsden. I'm an associate professor here in the Department of Biological Sciences. And since Natalia first started her faculty position here in 2022, she immediately made deep connections with many of the folks in our community, both here at NC State, but also more broadly around the Triangle. And so we're going to hear from a few of those many colleagues and collaborators and friends in the next session. And so all of our lives and our careers have been changed and shaped by our interactions with Natalia. And so I'll start with my story. So I first met Natalia on paper. I was chairing the search committee back in 2021, and we were looking to hire 2 new neurobiology colleagues. And I still remember being immediately struck by her application and just the clarity of her writing, as we've heard a little bit about already, and the passion and energy that just jumped off the page. You know, the excitement that she had about her projects and the clarity with which, you know, she could describe them. And Natalia was working at the interface of neuroscience and the immune system, you know, notoriously the 2 most complex of systems. And together, you know, that is, you know, makes them even more complex than the sum of their parts. And yet, in just a few pages, she was able to distill a unifying theme, linking stress, immune activation, and changes in brain function and how that impacts mental health through these key cells that we've already heard so much about, you know, these mast cells. And so I know that the whole search committee was really excited at the possibility of having a new colleague with Natalia's expertise who would be able to bring together and partner with so many different folks across our campus in neurobiology and toxicology, genetics, chemistry, veterinary medicine, and many more. And I really will never forget Natalia's chalk talk, which is, again, notoriously the hardest part and most challenging part of a faculty interview. It's sort of a black box that no one ever really knows what happens in the chalk talk until you do it. And Natalia's was fun. It was fun. Like, we had a really good time. Like, it was a conversation where we were throwing out ideas, talking about possibilities and connections. And she was so prepared and so poised and so open to new ideas and questions. And I think, all of us were really, having fun in that conversation. And so both Natalia and Krista Baker, who you're going to hear from in a moment, they both made our job on the search committee really easy. Their sustained excellence at every step of their careers, and their palpable excitement and passion for science and for training and supporting new scientists. That really made our jobs easy and made them easy choices for us to have as new colleagues. And so when Natalia started her faculty position with us as an assistant professor, she has had a lot of success in just 3 short years. She built an incredible foundation of success. She recruited 4 graduate students, 2 of whom we'll hear from in a moment. And she's trained 2 research technicians and provided research opportunities to so many undergraduates, I think I have lost count. She really had a passion for undergraduate research and saw that as one of her primary missions, to help provide those opportunities that were available to her and who had supported her career in her PhD and postdoc work. And Natalia immediately established collaborations with many of us in biological sciences, but she also collaborated with faculty in many other departments, colleges and universities, and some of those collaborators are here with us today. I'm one of those collaborators, and I had so much fun thinking about how my lab and Natalia's lab might be able to come together. And so my lab does very different work. We work with larval zebrafish, which are a really great model for doing things, when it comes to understanding the brain and behavior. But it's a totally different ballgame than working with mice, which was where Natalia's expertise was. And so, we decided that we wanted to partner and take advantage of using larval zebrafish to try and understand how mast cells develop. And so we partnered with another colleague of ours, Dr. Jeff Yoder, who's an expert in the immune systems of zebrafish. And we were fortunate to get some funding from the Comparative Medicine Institute here at NC State to try and establish a zebrafish reporter line that would allow us to not just visualize, but also to manipulate the mast cells in the zebrafish. And that, I can still hear Natalia's, excitement about this project, that, we would be able to use the advantages of zebrafish to be able to look in a living vertebrate animal and watch those cells develop, watch them emerge and migrate to their tissues of interest, or the tissues of residence, and see how they contribute to things like behavior, which is what my lab studies. And so we had some very exciting initial successes with our first pilot experiments. Chris, my lab manager, who's here with us today, I think we'll never forget how excited we were when we saw some of what we are pretty sure really were mast cells labeled with green fluorescent protein in the brain of a baby zebrafish. And Natalia was so excited, as you might imagine. She was jumping out of her skin with excitement at the possibilities that this might open up for us. And over the next several months, we worked really hard to try and establish that as a stable transgenic line. And as things go in science, unfortunately, we haven't had success in doing that. But as we continued to push forward with that project, Natalia was a constant source of ideas and encouragement. And that her excitement was really infectious and is still carrying us forward in working on this project. Natalia probably said yes to too many things. I think I probably advised her to say no more often, but I think she didn't like no as an answer, and so I don't think she liked giving no as her answer either. And somehow she had energy and passion for all of these projects. And she had the ability to do them all with excellence. And I really don't know how she did everything that she did. She was invited to give seminars at multiple international conferences and at leading institutions. And she's established ongoing partnerships with colleagues in other institutions. Her lab's first publication was accepted for publication in October, which is a huge milestone and something that every new principal investigator is going to be so proud of. And I know Natalia was exceptionally proud of that work. Her first big grant to the NIH was given a fundable score and still has a chance of being funded. And that is a massive, massive achievement. That is a acknowledgement of the excellence and the success of her vision and the groundwork that she laid over many years. And when it came time for Natalia to go up for reappointment as a second-term assistant professor, we had our faculty discussion, and that discussion really was mostly patting ourselves on the back for having done such a brilliant job in hiring Natalia, and Brian shared with us some of the thoughts of the faculty in just how impactful her work is, the brilliance of her writing. I think her writing, I have learned from her writing. I tried to be on the mentoring committee in helping her workshop her grants, and I think I learned more than I offered to her. So Natalia and I shared many things in common in our lives outside of science, like deep interests in art and music. Actually, right at the introduction earlier, we heard Natalia singing. So she was an incredible, but I think sometimes maybe a reluctant musician to be too forward about it. So we shared those deep interests in making art and in making music. But I will maintain that running is terrible. I'm not a runner. Running is terrible, cycling is better. And I really wish that I could have explored making more art and music with her. But I am so terribly grateful for all that we did share. Late nights in the DCL hallway, where she and I would always be the last ones there. And we were playing chicken, see who was going to go home first. It was usually me. Dreaming up new courses that would inspire our students and ourselves with new perspectives. Sharing exciting news about our grants together when no one else was around. Looking at brand new data and trying to figure out what it meant. Those were the best. Those were the best times. And when I think of Natalia, I will just always hear her voice saying, "That is so cool." That was just her constant refrain. And her unending and unrestrained excitement about science really was contagious. And it reminds me of how lucky we are as scientists to be able to follow our passions and to work in the discovery business. It's a frustrating and definitely an uncertain business. And Natalia knew that, of course. And yet she would not be deterred. Her persistence was rooted in her passion. And her passion was to make the world a better place, little by little, with her talents, of which she had many. In our scientific lives, Natalia and I were both deeply interested in how events early in life can have lifelong impacts. In neuroscience, we call this plasticity, the ability to change, which is perhaps the defining feature of the brain, that its structure and function can be changed by our experiences out in the world. Sometimes those experiences are hard, perhaps even devastating. Sometimes they're ecstatic, but they can completely change our perspectives and last a lifetime. I'm so grateful to Natalia for giving me this most amazing gift of being changed by her. And so with that, I'd like to pass the microphone to Dr. Maria Rogers, who's an assistant professor in the Department of Biological Sciences and an expert in the immune systems of aquatic species. She worked closely with Natalia, particularly in designing and teaching a new course on neuroimmunotoxicology. So please join me in welcoming Dr. Maria Rogers.
Hi, everyone. So thank you all for being here. And I really want to especially thank Kurt and Krista and John and Gretchen for all of their hard work in helping put on this event today, and so many other people. But if we could just thank them again, I think we all really appreciate it. So for those of you who don't know me, I had the great honor and privilege of co-teaching, actually creating and co-teaching, a course with Natalia here at NC State, Neuroimmunotoxicology Across Species. And we taught that course together in fall 2024 and also in fall 2025. So I was thinking about, what to tell you about her teaching. It's kind of a hard thing to talk about, right? I can tell you about the courses that she taught, and I can tell you about the students that she had and all of their amazing evaluations, because she was incredible in the classroom. But none of those things really like capture the essence of who she was as an instructor. So I started thinking about how I can put all of these things together, and I decided that maybe what I could tell you about are sort of the lessons that we learned from her and the things that we took from her during her time with us, and especially teaching with us. And I think we've actually already heard about some of these today, and I think if you knew Natalia at all, whether in research or teaching or just as a friend or family member, all of these things will ring true for you too. So the first lesson that she kind of left us with was don't dull your shine. So if you spent any amount of time with Natalia, or even if you just saw her across the room for 2 seconds, you would know that she loved to wear bright colors, fun patterns, big earrings, and that might sound trivial, but I actually think it was really important, especially for our students. So after she passed, we had been actively teaching a class together, and the students asked me, can we take a class and just talk about her? We have a lot to say, we want to remember her, and that's what we did. We shared stories and we shared memories, things that made us laugh, and several students all brought up the exact same thing, which is that they loved her wardrobe and the things that she wore. And a few of them were telling me that when they came to NC State to interview, they met her, of course, she was part of the program, and they were just kind of captured so much by what she was wearing, and she was so friendly and outgoing, and they all told me that it made them feel a lot more comfortable during this really stressful interview process, right? You have someone who's dressed in all these bright colors, is just comfortable being who they are, and is happy and telling students all these stories, and it's okay, don't worry, I know it's stressful, but you'll be great. And so that was just such a fun thing to hear. And then a student more recently told me a story about how they were going to a conference and they weren't really sure what to wear, and they had this outfit that was super boring and safe, but they didn't feel that comfortable wearing it. And so they told me yeah, at the last minute I just decided, you know what, I'm gonna wear what I want to, and it was very brightly colored. But I thought about Natalia in that moment, and I was thinking she was a rock star scientist, as we all know, and she was taken seriously, even though she dressed in bright colors and patterns and earrings. So it made this student feel more comfortable, I can be taken seriously too and still do those things, right? They don't have to be mutually exclusive. And so I think that's just an incredible lesson, don't dull your shine, be yourself. And that part of her personality is something that has already been intertwined with students and the things they've learned from her and how they kind of go out into the world and present themselves and interact with people. And I think that's really incredible. So second lesson, as it's sort of been alluded to, is say yes first and ask questions later. And this is the actual origin of how our class together started. So the toxicology program was asking faculty if we would be willing to pitch some courses that we might be able to teach, because they wanted to kind of increase offerings for graduate students. So I pitched the idea of, hey, what if I taught an immunotoxicology course, but it was across different species? And unbeknownst to me, Natalia had pitched a neuroimmunotoxicology course. And so neither of us knew that the other had pitched something, we just hadn't talked about it specifically. So a couple of months went by, and we both got contacted by the program saying, hey, you both kind of had similar ideas, I think there's some overlap here. Would you be willing to work together and create a new course together that kind of captures both things? And we said immediately, yes, we would love to work together. But the thing you have to know is we weren't really sure how this was going to happen because we were in a unique situation where Natalia and her lab were here, and me and my lab are at our coastal campus, which is like 2.5 hours away. So I remember talking to her and being like, okay, yes, I'm super excited, but how does this work? How do we do this? And she just went, oh, I don't know, we'll figure it out, right? You know aquatic stuff, I know mouse stuff, it'll just happen. Don't worry about it. And I was like, okay. And a new course was born, just like that. And the fun part about teaching with her is that we kind of tried to take that say yes first, ask questions later into the classroom too. So sometimes students would come to us and be like, hey, can we talk about X, Y, Z? And we both just go, yeah, sure. And then after class, we'd be like, do you know anything about that? No. Do you know anything about that? No. Okay, well, we'll figure it out for the next class, right? So it was a really fun way to teach and also learn together. Lesson 3 you've also heard today, which is collaborate, collaborate, collaborate. And that was a huge point of emphasis for her in her research and her teaching. So raise your hand right now if you had collaborated with Natalia on something or you were going to. So take a second and look around the room. That's a lot of people, right? Yeah. And I think I can probably speak on behalf of everyone when I say I really loved collaborating with her. She was an incredible collaborator. And I think for me, the hallmarks of collaborating with her is that she wasn't afraid to bring all of her ideas to the table, but she also wanted you and everyone else to bring their ideas to the table too. And she didn't say oh, my ideas are better, or I'm going to put them above yours because they were my ideas. She wanted to truly look at everything on the table and say, okay, what goes together? Can we put these things together? Can we put those things together? This is a good idea. We need something else. Let's keep that to the side. And that's how we worked in collaborating. And so in the second year that we taught the course, we ended up having 12 students, which was way more than the first time we taught the course. So we were thinking, okay, the way we taught it the first time with such a small group isn't really going to exactly work the same way. We need to change some stuff up. So we thought, okay, let's break our course into these 4 topics, and then we'll assign students into groups that are those 4 topics. And we tried to be very cheeky about it and say okay, what students don't really know each other? What students are from different programs? What students have different interests? And can we put those students together in groups and kind of model collaboration for them? Hey, we both do really different things, but we're still working together, and we're making something that's new and fun, and maybe you all can kind of work toward that too. And it was just fascinating in the end to see how students came together and worked, because a lot of them didn't know each other outside of class. But once we split them into groups with their topics, they kind of naturally formed this is kind of what I'm interested in. What are you interested in? Okay, do you want to do that thing? Maybe I can do this thing. You can do this other thing. And so in the end, they all worked together, figuring out their own strengths, their own interests, and just made a product that they could all be proud of, which was so fun to see because collaboration is I think kind of the bedrock of being an academic. And for me, it was just a real privilege to work with someone who believed in that as much as I did. And the final lesson for today, which I think is the most important one, is be a friend. And you might be wondering exactly how that relates to teaching, but I assure you that it does. So after Natalia passed and we had our class where we got to talk about her, we shared a lot of memories, and we shared funny things that happened. For example, we learned that several of us, independently, all had the same experience where Natalia was driving a university van and then lost the keys. But don't worry, we found the keys through the power of collaboration, so all's well that ends well. But one student said something to me in private that it just stuck with me, and every day since then I've thought about it. They were offering me their condolences, and they said this exactly. They said, "I'm sorry, I know that she was your friend too." And they didn't say, "I'm sorry, I know that she was your friend." They said, "I'm sorry, I know that she was your friend too." And I do want to point out that, sorry. Thank you. This wasn't one of her own graduate students. It's not a student for whom she was serving on their committee. And this was the only class that this student had ever had with Natalia, right? Like, that's primarily how they knew her. And yet they considered her a friend, even after that short period of time. And I think that was really impactful, because she wasn't just an instructor or just a researcher, she was all of our friends. And not just to those of us at her level, but to everyone on every level. And I hope that's kind of a lesson we can all take with us, right? So when we kind of go out into the world, we will promise ourselves we're not going to dull our shine. We're going to say yes first, ask questions later, which will lead to some hilarity. We'll collaborate as much as we can, and most importantly, we'll be a friend. Thank you.
Okay, so our next speaker is Dr. Krista Baker. Dr. Krista Baker is an assistant professor in the Department of Biological Sciences. She and Natalia were both hired as part of the same search as I alluded to a moment ago. And I was so thrilled and happy to see that when they started together at the same time that they immediately became fast friends. And not just friends, but colleagues and champions for each other's work. So, Krista.
So as Kurt mentioned, Natalia and I were hired together here at NC State, and we started on the exact same day in January 2023. But I first met her even before then. So as Kurt mentioned, the job ad that we both applied to was explicitly for 2 neuroscientists. And honestly, that was part of what attracted me here. Like, you're starting a lab, it's this big adventure, it's this huge undertaking, but I would get to have a buddy, a partner, someone going through the exact same things at the exact same time as me. Life in academia can be tough in so many ways. Having to live far from family. I never had to live as far from my family as Natalia did. Moving every few years and uprooting close friendships. Having to sacrifice time with family and friends for long hours in the lab. On that backdrop, having good people around you who understand what you're going through means everything. So as I was interviewing at NC State, I was dying to know who the other neuroscientist was going to be. While I was still negotiating my job offer, I saw Natalia's happy post on Science Twitter, when that was still a thing, that she had accepted an offer at NC State. And so I immediately ran to Google and trying to find her email address so we could connect. But she was hard to find. Her email address was not public. So I had to wait a few weeks until our head at the time, Carolyn, emailed both of us. And so instantly, I snagged her email address and I emailed her. And so we set up a Zoom to talk to each other. And I have to say that after talking to her for maybe 10 seconds, I was so relieved. Because I think, as a lot of us know, in science, there can be a lot of, how can I say it, jerks sometimes, probably like everywhere. But it was immediately clear that that was not Natalia. She was so warm, open, inquisitive about my science, and above all, passionate about her science. And who better to embark on this amazing journey, this hard journey with, than someone like that? So later that same day when we Zoomed, she sent me this message, giving me her phone number and saying that she was looking forward to starting this journey together. And that was the beginning of our friendship. So once we all arrived in Raleigh, all meaning me and my family, my husband and kids, and Natalia and Trevor, I quickly learned that that early introduction to Natalia was true to who she really was as a person, a scientist, and a colleague. Okay, I think we lost a slide somewhere. But let me see if I find it, because this, yeah. Okay, I don't find it. Anyway, it was a picture of her with my daughter, and then a few years later, a picture that my daughter had drew of her, and it was like a 4-year-old's drawing, but imagine someone with a ponytail, a high ponytail, and these big dangly earrings. And so she really, even in a child's eyes, those were the things that stuck out to her. And she drew it after Natalia's accident, and she said, Mama, here's something you can remember Natalia by. So with Natalia's camaraderie, my first few years at NC State were spent celebrating birthdays, holidays, basketball playoffs, go Pack, and just a sunny Saturday together. I curated a set of vegan dessert recipes and kept vegan butter in my fridge just in case of a last-minute barbecue invitation. And let me just say, there are very few people in this world who would get a Midwesterner like me to make vegan mashed potatoes on Thanksgiving. That's just how special Natalia was. As a colleague, I think we've heard from everyone that Natalia was generous with her time. Starting a lab requires so many things, recruiting and hiring people. And Natalia helped me interview many of the first people for my lab. She would take the time to meet with them, give me her impressions, and she shared her hiring philosophy with me, which is that anyone can be a scientist as long as they have the right attitude and are committed to working hard and learning, and that was all she was looking for. She gave me feedback on multiple grant proposals, and even though, so I am a neuroscientist, but I study hearing and fruit flies, which is so different than what Natalia does. And even though we were working on very different things, she always had important insights to share and important feedback that made my grant proposals better. But more than that, her enthusiastic support and her hype helped me keep going in the face of many rejections. And I want to highlight that she invested all of this time in my lab while she was doing exactly the same things for her lab, so basically doubling her work. All who know Natalia experienced her drive to help others. Her heart was limitless. She believed in the best in everyone, and she wanted so much to make things better for her students, for other students at NC State and elsewhere, for her research animals, as Brian told us, for the planet and the environment, for everyone who emailed her with tough situations. Over coffees, lunches, and beers, we talked about long-term dreams of growing neuroscience at NC State, developing new classes, improving support for students, and leaving our department and our research fields better than we found them. One quality out of so many that I admired in Natalia was that she was motivated by collaboration and open-mindedness. Do you sense any themes today, right? Like, I find it amazing that we kind of all honed in on many of the same things about her. She was motivated by collaboration, not competition. She was always connecting people and ideas. For example, when my first NIH proposal was assigned to the wrong institute, she immediately connected me with Brian Traynor because she knew that, like her, he would want to help. She also connected me with Maria, who just spoke. Since Maria is stationed out at the coast, we were hired at the same time, but I had never met her. But Natalia knew her through their work with the toxicology program. And so the three of us started texting, and we started meeting regularly over Zoom. And this trio really became a support group for us all as we figured out these new jobs, especially in the face of the grant and the immigration upheaval after the last election. At one point, Natalia, Maria, and I even started drafting a collaborative grant together, incorporating immunology, brain function, and behavior. There were many points during this drafting where it was hard for me to see how we were going to kind of find middle ground amongst these disparate research programs. So Maria's immunotoxicology in marine species, Natalia's neuroimmune interactions in rodents, and my auditory processing in fruit flies. But Natalia was persistent, and she was like working on that draft, and she was convinced that we could do it. So ultimately, it was like a limited proposal. We didn't actually get things in on time, and kind of life got in the way. But that gave me a firsthand knowledge and insight into how her mind worked. She was totally open to new ways of thinking. And that quality, her open-mindedness, was central to who she was as a scientist. To break new ground, you can't be tied down by existing ways of thinking. You can't limit yourself to what you have been taught about how things work. Natalia was forward-thinking and not afraid to take chances or to follow the directions her data led her. It's no secret that Natalia loved exercising, and after having a second baby, I was looking to get back into some exercise. And so true to Natalia's supportive personality, she offered to exercise with me. So we settled on bike rides on Sunday mornings. We would ride for hours and just talk about life, about family, but mostly about science, about our labs, about what was working, about what we were struggling with, about how from our two very different fields within neuroscience we could find ways to collaborate. On these rides, we would kick around all kinds of pie-in-the-sky ideas, getting inspired by each other's work and hyping each other up. And since we were on bikes, we didn't have to worry about getting pulled back into reality by a quick PubMed search. For example, whether fruit flies have mast cells. Spoiler, they don't. But a frequent theme of our conversations was how lucky we both felt to finally get to do the job that we have been training and sacrificing for so long. During our last bike ride, only a couple weeks before her accident, she told me how proud she was of her lab. Experiments were working, data were coming in, a paper was published, and things were running smoothly. She and her students were all on their way together. When we lost Natalia on both a personal and a professional level, I lost a buddy, a partner, the person who would text me at 7 a.m. to troubleshoot her latest experimental design, the person who loved playing with my kids and welcomed them into her house no matter how many times they pulled her cat's tails, and the person who would never, ever, ever commiserate with me over anything because she had no capacity for pessimism. Every failure I shared with her was met with certainty that the next time it would work, the next grant would get funded, that things could only get better. Natalia's life was shorter than many, but I remind myself often that her life was also longer than many. I take comfort in knowing that her last years were filled with accomplishing her goals and dreams, like starting her lab, publishing papers and book chapters, training students to pursue important questions, teaching new courses to spread her fascination with neuroimmune function, spending time with her family in Chile, and of course, finding her love, Trevor. As John said at the start, Natalia burned with an inner fire that she shared freely with all who knew her. How lucky are we all to have a tiny piece of light from that fire to guide us through the darkness we feel in her absence. Let us use that light to take new chances, make new discoveries, and honor the legacy of our beloved Natalia. Thank you.
Great, thank you, Krista. So next I'd like to introduce one of Natalia's graduate students, Hannah Lee Dixon. Hannah Lee just finished her second year in the toxicology PhD program here at NC State, and she's been working in Natalia's lab to understand how mast cells might mediate the effects of toxicant exposures.
Thank you, Dr. Marsden, and good afternoon or maybe good evening, everyone. As Dr. Marsden said, my name is Hannah Lee, and I was fortunate to be one of Natalia's PhD students. So I joined the lab in December of 2024, and within the span of just a year, she left not only an indelible mark on my professional and research career, but also on me personally. So it's an honor to stand before you today and reflect on what she meant to me and to so many of us in her lab. So as I tried to begin this speech, I kept finding myself at a loss for words. And even now, I'm not sure that what I've written is exactly the perfect thing to say, though I'm certain in Natalia fashion, I could talk for hours or more about my research and my experiments. But today, I'll spare you that granularity of those experiments, because Natalia was more than that, research and professional mentor for me and for everyone in lab. On the outside, Natalia was someone always walking, always taking the stairs, always reading papers, always ready for the next amazing experiment, and always, always bringing up mast cells. But underneath her fast-paced strides and unending vitality, Natalia was a confidant, a friend, and one of the few people I knew who could discuss the meaning of life and experimental design and the best beer to drink while painting, somehow within the span of just 10 minutes in her office. She was the face you scanned the room for during a presentation, and she was always waiting one glance away to smile and tell you just how amazing you were. And you knew that she meant it in more than in a professional and academic way. She was also the PI with 200 open tabs, frantically searching for this one article I read at 4 a.m., while also being the best friend you knew you'd have once you graduated. As graduate students, she was everything to us and even more, the epitome of joy, kindness, and brilliance. And for me, she was also the embodiment of one of my favorite quotes by Luciano de Crescenzo that says, "We are each of us angels with only one wing, and we can only fly by embracing one another." And Natalia was just that, an overflowing cup of passion and creativity who took pride in helping those around her also take to the skies. It is because of her I found my home at NC State, and it is because of her I now know how precious every moment we all get together really is. Of the many memories I have of Natalia, few can outshine the first time I met her when she was sitting at her desk surrounded by a collection of empty energy drink cans. Her smile was so great and so joyful as she was talking about her research, and I was nervous as ever, and I remember she started talking 1,000 words a minute, and if you knew her, you know just how fast she could talk. And something about it was overwhelming in the best way possible, and if I took anything else or nothing else away from that meeting, I'm certain she said the words mast cell over 100 times. And I was stunned, and at the time I thought her mountain of energy drinks was the culprit. So I innocently asked her, "Have you had any caffeine today?" And she said, as if I was crazy, "No, I'm not even awake yet. I'm just on my second energy drink of the day, and I've got like 2 more to go." I was a little worried at that point. To this day, I have never seen someone consume so much caffeine and be functional. And to this day, I am in awe that that energy I saw in my very first meeting with her was the energy she had in every meeting that followed. And it was just her, because even without caffeine, every atom of her being seemed to vibrate with a passion for research and for connection that eclipsed even the brightest and most determined in the field. After she passed, Natalia's relentless energy somehow continued to surround us and showed up in our everyday lives. Elevators not working just when we didn't have time for the stairs, mice going haywire for no apparent reason, and life in general taking on the sporadic and fast-paced nature she always had. But nonetheless, as each day passes both slowly and so quickly, these are the reminders of her I cherish because they tell me that she still surrounds us. And now, as I find myself randomly opting to take the stairs and also preparing for the busiest summer I could have imagined, I know this would have never been possible without her. She lit the spark and kindled the flame, and it's such an honor that some piece of her passion for research and for life in general lives on in me and in my lab mates. And even more, it's the depth of her presence that I still feel now that makes me wonder if she'll ever just walk back in one day. It's the thought I cannot yet seem to shake, and every day I still expect to get a text or an email asking for an update on data, mice, or my husband's jaw surgery that she could never remember at the time. And standing here today with all of you, I now like to think that she does in fact walk in, that she stops by our offices in the lab and finds us still procrastinating for probably 1 too many days and stressing about minutia that inevitably doesn't matter next week. And I like to think that she'd be so proud in that moment, not because we've carried on in her honor, but because we've done it all while shouldering such a tremendous loss. And she'd see that she taught us more than research and technical skills, and she'd see that we learned how to live, how to laugh, and how to love in even greater capacity simply by having known her. Yes, she would see that some of us have developed affinities for caffeine that might rival hers now. And yes, she'd see us waiting with open arms. But she'd also see each of us lending a wing to one another, embracing through all of it so that we might still take flight just as she had hoped. For me, she is in every mast cell and in every energy drink. She's in every stairwell I take and in every graph I generate, and how thankful I am for all of that. May we all try to live with the passion, kindness, and vitality that she did with such ease. And may we all know and feel so lucky to have been so changed by knowing her. I know that I do. Thank you.
Thank you, Hannalee. Our next speaker is Jared Franges. Jared is a third-year PhD student in our biology PhD program, and he's been working in Natalia's lab to define differing roles of mast cells that originate in different tissues. Jared.
Thank you, Dr. Marsden. So yeah, as Dr. Marsden just mentioned, I'm a third-year PhD student in biological sciences, and I had the distinct honor of being one of her first grad students along with Yang. And as we probably all experienced, choosing a grad school is a pretty stressful time, especially when you get waitlisted by quite a few. I won't name names. But I had a very good relationship with my undergrad advisor, and her advice was, while the research topic is pretty important, you want to like what you're doing, even more important is the advisor, and if you could see yourself working well with that advisor. So, fast forward to my first meeting with Natalia. She pulled up to the hotel on a bike, no surprise there, and we sat down at lunch after talking a bunch of science, of course, and she asked, "So Jared, do you like beer?" And I said, "Oh, yes, I do, Natalia." And that's when I knew I was in the right place for sure. I'm pretty sure I texted my undergrad advisor about an hour after that and said, "I think I found my place." So knowing no one here, having no family in Raleigh or North Carolina, I took that leap and came down here to do some work with Natalia. Also, that visit, I spent the rest of it trying to keep up with her, both because she walked quite fast, and also because how much science I was getting thrown at my brain, and so both of those things, I was pretty fried after that day in the best way possible. So fast forward to now, I feel like it's pretty clear from everyone's talks that she had accomplished a lot in a short bit of time at NC State, and I'm honored to have been a part of that. I feel like I only got a very small blip of how great she was, and I'm still very grateful for that. People have mentioned our first publication as a lab, and I'm even more honored that I was able to be the first author on that and kind of lead the charge on some of those experiments. So in honor of Natalia, I'll throw a little bit of science at you. So that paper, the principal finding is that an environmental contaminant, which is a banned class of flame retardants called PBDEs, they got phased out in the early 2000s. However, they remain pretty pervasive. And basically, developmental exposure to those contaminants actually blunt mast cell responses. And funny enough, when her advisors from Michigan State were speaking about some of her earlier findings, I now have even more questions about some of the things we found, and of course I think she probably wanted me to have more questions. So yeah, we had a few experiments, and of course mast cells, I could say it 100 more times if we wanted to, were one of Natalia's primary interests, and we were very interested in how a contaminant could affect their function along with how mast cells can modulate behavior. That's a pretty core theme of what everyone's talked about so far. And our first finding that we kind of had that eureka almost was that the mice exposed to these PBDEs, they had a blunted hypothermic response if we induced this passive systemic anaphylaxis, very similar to some experiments that were shown earlier. And Natalia was super excited. I mean, I couldn't contain the energy in that third floor, mouse room. I'm sure everyone in that building knew that we were onto something. So after that, we ran numerous experiments. This led to experiments in bone marrow-derived mast cells, which she would be happy to see me at the hood doing cell culture, and that's where I was probably most happy. And we took cells exposed from those mice, and those also exhibited hyporeactivity, as well as differences in some of the granulation and other markers that kind of give us clues on how they function. We followed these up with experiments like RNA-seq, where we actually found some sex-specific changes in transcription, and we also had some behavior experiments that showed some sex-specific changes in anxiety-like and social behaviors, all things that I think really nicely wrap up all of her interests that were mentioned today. And I didn't really fully appreciate that at the time. And I think after hearing everyone talk today, I appreciate that even more. So I think that's great. And, you know, I spent about 2 years working on this, and those 2 years were probably some of the most difficult and also rewarding times. The writing process, especially with Natalia, is difficult. I'm pretty sure I drove her crazy. We had to respond to some criticisms from reviewers, and that required me to run quite a bit of qPCR, which I may or may not have saved for one weekend. And I got a pretty stern email from her on a Friday that said, "Jared, where is this data, and what are you doing?" So, naturally, I spent 12 hours a day in the lab on the weekend, which I actually kind of enjoyed. And I ran into her, and she was bringing slides up from the other floor, and she wanted to image those, and she goes, "Man, I feel like you've been here a while. You should probably take a break." And the look on my face was probably hilarious because I had just gotten that email that was like, "Jared, where is this data?" So, you know, either she forgot she emailed me that, or she just really wanted me to take a break, or something. I don't know. It was a test, I think, maybe. So, you know, with that in mind, I'm sure she would have been pleased to know that I may or may not have written this speech last night over a beer. So all jokes aside, I do really cherish the time I spent with her in the lab and, of course, outside of the lab. Dr. Rogers actually talked a lot about that class, and I was in the first iteration of that class she taught. And we took a trip to CMAST, which was awesome. Getting to know her outside of the lab was really special. Even when apparently we all have van stories, even when we went half a mile down the road without the headlights on at night, that was fun. The lab parties were epic. Getting to know her outside of the lab there was also amazing, and I'm lucky that I was able to do that and that we all were. And also going out to a brewery, get a beer, also disguised by more science. That was actually Science on Tap from Dr. Baker's husband, which was a really good talk, actually. But it just showed how much she loved science, even over a beer, of course. Even better over a beer. So yeah, I know I wasn't as eloquent as my lab mate, but Natalia, again, as we all know, was an incredible scientist and an incredible mentor and a friend. And coming directly from undergrad, I had a lot of learning and growing up to do, and Natalia was an amazing teacher, especially with that in mind, just kind, patient to a point, and brilliant, of course. So I attribute a lot of who I am as a scientist, a learner, and just as a person in general to her mentorship. And also getting to know her and Trevor over the past year or two has been amazing, and I hope Trevor also realizes how much him and Natalia have meant to me over the past 2 years. But yeah, thank you.
Thanks, Jared. So I'm honored today to introduce our next speaker, Dr. Heather Pattisall. Dr. Pattisall is currently the Scientific Director of the Translational Toxicology Division at the National Institute of Environmental Health Sciences. Prior to moving to NIEHS, Heather was a professor in the Department of Biological Sciences and the Associate Dean for Research in the College of Sciences. She and Natalia shared many interests, and that brought them together both as collaborators and friends. Heather.
We did, including running, which is still better than cycling. I'm sorry, but you know, we'll just have to, we'll see. But yeah, it's been really fun and bittersweet to learn and hear about Natalia from her science family. She's impacted all of us for the better, and it was such a privilege to get to know and work with her. And so I wanted to close this session out by sharing some of the work she did with my group and show you how it ties in with the work she did with Brian and Adam and AJ and the others. And this is the chemical, Firemaster 550, that John Meitzen mentioned at the beginning of this talk. It's the chemical that we were working on when she passed away, right after we were working with PBDEs. And I really like the bottom one, the TBH, because it looks like a crab. And she drew that one on my board at one point and drew all sorts of little funny things. And I can never look at that chemical without thinking about how Natalia turned it into a crab. But I was really excited when I got the opportunity to move to the NIH, because I'm like, oh my God, it's the NIH, right? And I had this like moment of imposter syndrome too, of like, oh my God, it's the NIH. But I was really excited because NIHS is one of the only institutes that's not in DC. NIHS is actually 15 minutes down the road. And I was like, well, that's awesome, because I can still collaborate with Natalia. So we kept talking about how we were going to keep everything going, even as I moved my lab 15 minutes down the road. And so I think you've seen today how lovely and wonderful the people in this department are. And so they really allowed us to leave what we needed here at NC State to keep our work going. And that's what we've done for the last 2 years, is continue to collaborate with these tremendous people and do some of the work that you've learned about today. And so for a bit of history, I was part of the team that recruited the dream team, the dynamic duo, to NC State. And I still remember how remarkable that interview was, not just the chalk talk, but her talk in general. And I remember looking across the room and we all were just like, it was like a mic drop moment of like, we're done, right? Like, she's coming here. And what I remember is as we were leaving her talk, it wasn't a question of whether or not she was worthy of us. It was a question of whether we were worthy of her. And we just wanted to impress her at that point. Like, we were in such awe of her. I really felt like we were auditioning for her that day of like, is she gonna pick us? Like, that'll be amazing if we can come and get her here. So when she got here, I, like everyone else, bum rushed her door and was very easy, wanted to collaborate with her. And I was like, hey, you know what you should do? You should do toxicology, because toxicology is stress with chemicals. So it's way easier than doing all of this like defeat stress and all that. You can just give them this flame retardant, and then we can see what happens to your favorite mast cells. And she's like, let's go. So that's basically what we did. And so I've been working on flame retardants with another Heather Stapleton, who's in this room at Duke University for almost 15 years. And she's been one of the most fun and amazing people to collaborate with. And Natalia's fun and amazing. So I was like, well, obviously, we all have to collaborate together and make it extra amazing. And so when she got here, that's where she started working on PBDEs. And so Jared just introduced that paper. And so many congratulations to Jared. This is the paper. It came out in January. And what's really remarkable about this paper, you see how many authors are on there? This is what fun scientists do. We're like, let's get everybody in here and do all this stuff. 9 of those authors are students. That was Natalia. Natalia really wanted to support the work of the students. She wanted everybody together working on a project because she got excited about it. One of my mentors said something that stuck with everyone that worked with him, which is credit is infinitely divisible. And Natalia lived that. Credit is infinitely divisible. And it was fun to work with her because you got to work with all these incredible, fun people. And so congrats to Jared. Congrats to everyone in her lab who worked on this paper, Hannah Lee, Yang, who are here. This paper is huge for toxicology. It is going to be a landmark paper for toxicology because it's uncovered a completely new way that these chemicals can impact brain and behavior. And what's really interesting is PBDEs are one of these, what we call sort of legacy data-rich chemicals. Everyone's like, blah, they're boring now. We totally understand how they work. But no, she used this chemical to discover an entirely new way that chemicals can impact the brain and have multi-organ effects, not just in the brain, but everywhere else. And so this is going to be a landmark, incredibly important paper for toxicology. It's only been out since January and it's already starting to get cited. So congratulations to her lab on this accomplishment. And so of course, PBDEs are in the process of being phased out, and I was working on this mixture with Heather Stapleton, and so we said, okay, now we want to rope her in and try and see what this chemical mixture might be doing, like the PBDEs were doing. And it's really fun because if we're all, this chemical is used in furniture. We're all sitting in chairs. You're probably all exposed to it right now. And so if you look at the mixture, you're looking at what real humans are exposed to out in the world. But then you can also go chemical by chemical. And if you start to understand how those individual chemicals impact the brain, now I, as a toxicologist at NIH who talks to EPA, can screen for the ones that are causing problems. And we can start to make a real difference in human health and really start to figure out which chemicals that we need to get a better control of in the world. And so that's where Natalia's research is going to have impact for a very long time, is discovering these new mechanisms to try and understand how we sort through the chemicals in our lives that are making us sick and the chemicals in our lives that are not, because obviously we want more of the chemicals that are not. So that's what we were working on at the time that she passed away. And this is a paper from her time with Brian that we pay a lot of attention to, because he talked a lot about oxytocin in the extended amygdala and how oxytocin in the extended amygdala can cause that social vigilance. And so what we've seen with that flame retardant mixture that I've shown you, and I love that Natalia works on weird critters, because like we've all worked on weird critters. I also work on weird critters. So I work on a monogamous rodent. So these are rodents that mate for life. Both the mom and the dad show parental care. And if I expose those rodents to that flame retardant, the male no longer bonds. The bond is completely gone. And we've done it 3 times. We've done it in my lab, we've done it in John's lab, we did it more than once. We see it every time. And our females are hyper anxious. And they're hyper anxious, we think, because they're engaging in social vigilance. And so when we saw this paper and we talked to Natalia, I was like, oh my God, she's really going to help us figure out the why of what's going on with this chemical. And so we were looking at nucleus accumbens, because that's John's favorite brain region. And we were looking at medial preoptic area, because it's my favorite brain region. And then we read this paper, and we were like, now we have to look at the extended amygdala, because that's probably the right area. And so we did. And my lab texted me this morning that that paper that we worked on, where we did some of the transcriptomics in the extended amygdala, was submitted to Toxicological Sciences this morning. So I thank her for the inspiration to get that research done, and to Brian, sort of tangentially, for helping us understand some of the mechanisms from what's happening with these chemicals. And so it just shows the cross-disciplinary lasting impact that Natalia is going to have across multiple fields. And this is another one. So this one came out this month. So this is another paper from Brian's lab. So you have this effect in science. Even when you leave a lab, you're still part of that science family. And when your science family moves things ahead, the things that you inspired and the things that you created, you still are an author on this paper. And so I imagine many papers are going to come out in the next few years. Certainly from my lab, this will be true, that represents what Natalia has contributed and done. And this is one that came out just this month. I also wanted to highlight this book chapter I wrote. This gets me so excited. I'm like, I made a toxicologist out of a neuroendocrinologist. This is great. So this chapter that she published really sort of embodies her thinking about how these systems interconnect, both the immune system and the neuroscience system, in a way where she's also thinking about toxicology. And so we talked a lot about her art. This is one of the figures from that book chapter. And so you can see where, even though the chapter is really focused, in this case, on compounds that are found in cannabis, but you can see the flame retardants are in there. She's thinking about all these different cells, including the mast cells. And then in there, see the plastics in the flame retardants? She's also starting to think about plastics. And in the institute that I work, we talk a lot about plastics. And just in the last couple months, NIH has committed over $150 million to work on micro and nanoplastics. She published this a year ago. So she was thinking that far ahead. And you better believe, as I pull these programs together at NIH, mast cells are on the list. I'm like, but the mast cells. So I am going to be ruthless and relentless about making sure some of the things that she identified and discovered along the way are going to be included in those research programs as we try to understand how the pollution that we put into the environment may be impacting our health. And to close, as you heard from folks, she had put in a very competitive R01. And to put this in perspective, I think it's not understating that 2025 was probably one of the most unbelievable, catastrophic moments for science in US history. And so against that backdrop, for her to have a fundable score as early in the career that she has is extraordinary. That is an extraordinary accomplishment. And this is a summary of those aims, and I wanted to put this up here because this is the roadmap. This is how we keep her work going. This is how we make sure that her ideas live and breathe in the real world. This is what her science family is going to keep going. And so I wanted to put it up here because she draws amazing figures. Anyone that's seen my figures, you're like, oh, Lord. Natalia draws amazing figures. She doesn't just make beautiful art. She's not just an amazing writer. She's also an incredible illustrator. I've seen her explain science to everybody. It's incredible what she can do. And so this is a brilliant piece of work, and I think that's where it's headed. And she's got a lot of that Fire Master work in here, and I'm so proud, and I'm really committed to making sure that this work gets done. And so with that, I just wanted to close. I wanted to thank everyone, and I wanted to say, Natalia, wherever you are in this world, girl, we will miss you, we love you, we've got you, and we will make sure that this work gets done. Thank you very much.
Thank you, Heather. And so finally, it's my great pleasure to introduce Trevor Huffman, who's going to tell us about PBDEs and how mast cells work in the brain. Right? Oh, no, sorry. I'm kidding. Thank you. And Trevor is Natalia's fiance and a friend to many of us here in this room, and I'll leave it to you both.
Oh, that's a good one. As her lab knows, I was the support. Got the beer, I got the ice, I got the cooler for anything in the lab parties at our house. But I'm going to read a few things, if that's all right. Stick with me. If you guys have tears, let them out, because I know I'm going to. This is from Matthias, her brother. I have so many beautiful memories. Hold on a sec, let me get this screen brighter. I have so many beautiful memories with you that if I told them all, I'd never finish. I remember nights when I was 12, you were 17, in your room listening to Jorge Drexler, you reading me your writings, telling each other our problems, laughing until we couldn't breathe, and crying rivers together. I loved following you everywhere, and it was during that time that you started becoming my greatest role model, which continues to this day. Because of you, I started searching for depth and art and knowledge, questioning the existential, being in awe of culture, science, and nature. Your love for animals was always there. I remember the unconditional love you had for our dog, Yelcho. Like, what kind of name is Yelcho, Matthias? I know you're watching. Then he goes to say, you even told me you were going to marry him. You were going to marry Yelcho? Okay. Any animal or bug, no matter how threatening it looked, you'd pick it up, observe it, and let it be free. Because to you, every life was worthy of love. I remember nights in Payawente, looking at the stars, talking until very late. More than once, we slept out in the open, watching the vastness of the universe. When you left for the USA, our communication continued through endless video calls. And it was just like when we were kids in your room. We talked about philosophy, art, humanity. We cried, we laughed. Our connection was always incredible. My best friend, my shoulder to cry on, my confidant, my advisor. I don't know who I'll ask or share my scientific and philosophical questions with. The only thing I know is that we'll keep having our conversations, even if we have to break through time and distance. I do know there's a lot of scientists in here that will answer those questions for you, Mateus. So send Casey Wagner an email at casey.wagner@gmail. No, I love you, my sister, and I will admire you forever. I'll live my life thinking about what you would do. Thank you, Matthias and Danny and Lucci. I know you guys are watching. This is from Karina. She's the middle sister. Tati wasn't just my sister, but she was more than that too. We shared life like true soulmates, thousands of fights. I can believe that, thousands. Obviously sisters in every sense of the word, but also millions and millions of precious moments that I'll treasure forever. our fits of laughter until we cried when we sang in harmony on vacation trips. If you guys have never heard Natalia sing, oh, her and her sister, they can hit every note I can't. The craziness where I was the accomplice on those trips to other countries. And now for my little girls. Thank you. Taddy was a scientist aunt from the United States, an aunt who adored them, and they adored her back, even from far away. Always the cool aunt. She was an exceptional person, one of a kind. She left behind a tremendous legacy. I mean, all of her professional achievements, also the personal ones. We carry her mark deep within us, the way of being that resisted apathy, that never settled for accepting hatred as something inevitable, that loved people and nature so deeply. For as long as I can remember, you were my heroine, the one I had to follow because you were incredible, my role model in so many ways. Tati, I love you indefinitely forever. I hope we heal little by little, and that we always remember you, but not in our thoughts, also in our actions, to create a better world, one more like the one you wanted. You will live forever and be an example for me and my little girls. Love is eternal, it is invincible, it is deep, it will hold us up. It doesn't change with time or with disagreements or with distance, and it doesn't change with your passing either. Tati, you are a little further away, but you will always be here with us. I love you, my Duque. This is from Cookie and Juan Carlos. Everybody wave at them, they're watching. Oh, I gotta say, before I read this part, their family is one of the most amazing families. You go down there and you sit around a campfire, and they made a song for my brother Jeremy. Stand up, Jeremy. And they're so talented that they made a freestyle acoustic radio hit on the fly called Hera Mia, and I will never forget the melody that they sang, and I've never heard my brother laugh that hard. And this is from Cookie and Juan Carlos, whom I love dearly. There isn't a day, not a single minute, when we don't think of you. Sometimes we feel like you're about to appear, just like always, with a message, as if we could see your name on the phone and find you there alive, radiant. Our heart leaps, because our love for you is so great that it doesn't understand absence. We miss you with a force that words cannot hold. We can't believe that you're no longer here, and we feel lost. But even in this darkness, what we feel most is love. A love that doesn't break, that doesn't disappear, that has no end. We are so proud of you, my girl, of your strength, your sensitivity, your way of fighting, of your dreams, of your courage, of the way you loved life, even when life didn't always treat you gently. You were and you are an infinite light in our lives, and though it hurts more than anything, we want you to know that your light is still with us, that your name is our deepest affection, that your memory is a constant embrace, that we carry you here close, always. There are moments when we look at our phones and think you're going to appear, but then we remember and our souls break a little more. Not because you are no longer here, but because we still don't know how to live without you. We don't know, but we will try, step by step, because I know you would want to see us at peace. Thank you for being our daughter. Thank you for everything. Thank you for the love you gave us, which now lives in us like a treasure. We love you beyond everything, my girl, forever. Thank you, Juan Carlos and Cookie. Now from me. I don't know how I'm going to get through this. All right. Oh, I came to know Natalia's innocent spirit and curiosity through photographs her parents would send me. She would have a big belly, and her eyes would be shining. She'd always have food on her face. She had a missing tooth, which she also had a missing tooth as an adult. I don't know if you guys ever knew that. She'd come home and be like, "Where's my tooth?" Be like, it's in the plants. You said it in the plants. So when you guys told me about her losing the keys to the vans, I was like, oh my God. Every day of my life was I looking for her keys. You'd think that we had been smarter and got an air tag or like a little beeper on it, but I really liked the struggle. Oh man. But those pictures, they showed her spirit come out, and I saw pictures of her sister and her brother looking at her the way younger siblings do, wide-eyed and adoring. I saw proud photographs of her grandparents, Manfred and Kedden, watching their beautiful Duque Wilkins family legacy grow. I eventually met all of them in Chile, and we went down to Patagonia and we sat under the stars, and it's just this beautiful place. And that's where, we were going to get married in January. And Natalia was fierce. She had a wild, fierce heart, an insatiable need to understand how and why things worked. One of my favorite stories about her was she found a dead cat walking home walking to school in elementary school. And instead of doing what most people do, like I would have ran from the dead cat, she picked up the rigor mortis cat, carried it into school, and tucked it beneath her desk. Like it was no big deal. Just got a dead cat here. It's just a little stiff. We're going to figure this out. I think she thought she could save the cat and learn how to save the cat in elementary school. As you can imagine, this did not go over well with her very strict German school teachers. And I can almost hear them now yelling, "Scheiße, Natalia, Scheiße!" She was put in detention, probably suspension. But I never got over the sheer audacity of just that story of her as a kid picking up a dead cat and trying to save its life. I'm sure she would giggle and smile widely at me, graciously indulge me, just as she always did when I tried to make her laugh. The point is, she always wanted to do her part to save the world, no matter how small that part might seem. She wanted to give everything, man or woman, child, dog or cat, plant, flower, tree, wildebeest, stink bug, red-breasted meadowlark, a better, happier, healthier life. Natalia dreamed of a society, a culture, a humanity that cared for one another more deeply. To her, love was a verb expressed through the compassionate action in the smallest daily ways. And she lived her life as that kind of verb. She believed every living thing could have a richer, more meaningful experience if only we could muster the courage to act. This was Natalia at her core. She lived a robust, fully packed life of action, love, laughter, compassion, and I was so lucky to witness it every day. Natalia's life and later her scientific career became devoted to diminishing suffering through understanding. She wanted to know how her creative mind and scientific rigor could lead to discoveries that would improve life for all living things. Her mice needed better, more sociable, three-dimensional habitats. Her friends and family needed more art. Our home needed more plants and color. Her office always smelled of fresh eucalyptus, peppermint, orange spice. Who has smelled that and when she went to her office? She had the best smelling office. Every time I visited her there, she was furiously typing. Like, I was like, what? How do you type that fast? And then she'd be meeting with a smiling student or gulping down another C4 energy drink. My niece Ariana gave me a C4 energy drink, and I keep it next to a picture. I know one day it's just going to be cracked open when I come home. But yeah, she did love her energy drinks. There's no wonder she accomplished more before sunrise than I did in an entire day. Oh, man. This wasn't merely discipline or habit. Natalia was endlessly fascinated by the questions science asked of her. Some mornings before sunrise, I'd crack open an eye and find her scrolling through the latest research on mast cells or neuroscience or the ways environmental contaminants affect the immune system and brain. And I have never, ever heard the term FM 550 and mast cells so often in my life. Thank you, Heather. When we bought our first home, she picked out new chairs, couches, cabinets, armoires. Our house looked like an IKEA warehouse. I'm not kidding. And I can tell you this, I was not the one leading the charge when it came to reading instructions and assembling the furniture. No, I was not. My family will gladly tell you I do not do well with directions. And even when I tried to bluff my way through and I said, Natalia, I got this. It's okay, I got it. She simply said, move over, Bolo, hand me the Allen wrench. Things moved much faster that way. Her determination fascinated me. Her effort was off the charts. Her brilliance, her beauty, her empathy, it made me a better man. At one point, she told me to never call her baby. You know, I thought it was romantic. I'm getting married. Baby, I love you, baby. She goes, never call me baby. She invented a new term, bolo. I said, bolo? That's not a word. That's not a word in English. She said, yeah, but it doesn't mean baby. Within a week, bolo had become our word, a symbol of our love, our honesty, our shared weirdness. We each had our own styles of authenticity, but I loved hers most of all. Natalia loved to garden, walk, bike, run, lift, paint, sing, dance, cook, play with our cats, travel to home to Chile, see her friends, throw dance parties. She loved to laugh. She loved to laugh so hard that tears would stream down her cheeks, so hard that she rolled off the couch, so hard that she snorted oat milk through her nose multiple times. She loved making a house feel like home. At the end of each day, she loved to sleep. She loved to sleep with her pinky finger wrapped around mine, 17 pillows piled over her head to block out all sunlight and sound and light. This woman deserved more sleep than anyone I ever have known. And she loved our cats as much as she loved our big dreams. She adopted cats like some people collect cookbooks. And I was lucky to support, witness, and share in those dreams. At times, I felt like a coach. You know, I played sports. I played pro basketball. For God's sake, I can help her get tenure. Right, Bruce? So I would say the worst cliches: trust the process, stay positive, stay ready. You can't control the outcome, just play the next play. Bolo, you got this. I'm not sure it helped much. Managing your jump shot, it turns out, is way easier than managing a colony of genetically modified mice. But if I did one thing well, it was trying to make my Bolo laugh, relax, enjoy the moments. They were the most meaningful gifts I've ever been given, to be alive beside her, to create with her, to choose with her, to build something meaningful together. It was the best gift a human can get. We dreamed of spending summers in Patagonia country, becoming even more deeply woven into the growing Duque Wilkins family. She would have treasured watching her nieces and nephews, Anna, Lucia, Ines, Luci, grow. She would have loved returning to Michigan to see my family, to watch Lila, Ariana, Jack, and all the others grow in my family as well. To be surrounded by family, to witness the next generation. I know for a fact that would have been her greatest joy and triumph in life. Which is why I'm so thankful to have my family here, everyone that has supported me, sent me emails, the meal trains. Oh, I don't even know where to start. I'd be face down in the ditch without all the love and support I've gotten. I'm pretty sure of that. Hell, my mom practically turned around a cruise ship and threatened to swim to shore just to get to us the day she passed. So thank you, Mom. And still, grief remains. It rests in my bones. It mixes with my cells, my neurons, my soul. It is the immeasurable cost of a priceless love. But the burden of sharing our emotions will never outweigh the love she had for us. And so I have questions. What do we do without Natalia? What do I do without her huge heart, her strong bones, her curious eyes, her long toes, her hands in the soil, her beautiful mind? What do we do without someone so unique and compassionate and empathetic? I do not know your answer. I only know mine. I will do my best to honor her in everything she taught me about living, because her life was the very best kind of verb. And she is not entirely gone. Bolo is dancing with us in a different way now. I'm sure she has found answers to all the questions she's been asking. She's gone through 1,000,000 doors, and I'm sure she'll go through 1,000,000 more. The day she died, 5 red-breasted blackbirds came to see me. Her spirit animal, she told me. They just sat feet away, staring at me, blinking, and I felt a tingle of warmth in my belly and a pulsing of electricity in my fingers, just like the night I held her hand as she passed in the hospital. Thank you, Bob and Amber, for being there with me on that night, the worst night of my life. And I do not have the scientific acumen, although AJ says I do. I don't know if that's true, AJ. And I don't have a research lab like Natalia's and all the talent that she had there to prove that she still exists. But I am convinced that souls and the love you have for those souls does not die. It changes form. And if you ask her to show you a sign, she will find a way to send it. So I thank the universe for introducing us, for tangling our fishing lines in the Lansing water, for giving us the time to fall in love, because falling in love with Natalia was the best decision I ever made. She was by far the best teammate I've ever had. She was the purest kind of love, the purest kind of human, the purest soul I've ever encountered on this great round ball of cosmic dust and rock floating through space. And when I moved into her apartment in Bath, Michigan, she was working for AJ and Ken, hanging out. I just remember thinking, man, scientists are way cooler than pro athletes. What was I doing? And when we came to NC State, I felt the same. Even after she proposed to me in a white and turquoise RV, I knew I was in a different world. She was so brave, an eclectic artist, a true underdog that always followed her heart. It was as if our souls had always known each other. We simply had to clear away the older parts of ourselves to recognize it. And I'll finish with this. When she passed, I had her phone, and I went into it, of course, just to read and look at pictures. But I found grocery notes in her Apple iPhone notes, and there's hundreds and hundreds and hundreds. How many, did the lab ever get iNotes from her? iPhone notes? No? Okay, well, I'm going to read a few. These were just grocery notes, and they would read like this: avocados, dark chocolate for Bolo, IPAs, tofu, double dark chocolate ice cream for Bolo, onions. And then nestled between food items, a profound observation. Science is not a fact. It is an endless question. I believe in the quiet intelligence of all living systems. Mangoes, apples, almond peanut butter, soft toilet paper for Bolo. Oh, man. And as of now, we have raised more than $65,000 for Natalia's memorial endowment. Let's give her a hand. I'm so grateful for everybody that has given, supported, donated. It's just been the one tiny thing I feel that I could do, and I just am so appreciative from my heart to you guys. My friend Ash, thank you, Ash. He also has a website where we can buy her art. Her art here, this is a print, and all the proceeds will go to the endowment fund. And my friend Ash will mail you a copy. He works at an art store. It just worked out perfectly. So he set this up for me. And I'm just so thankful. Krista Baker, thank you for setting this all up. Love you. Want to thank the organizing committee. John Meitzen, Kurt, Dean Owen, Bruce, Gretchen, also Caroline and Tony. I don't know if they're here. Krista's husband, Casey, my first pickleball mate before he had his son Reed, and I never saw him again for a year and a half. To be expected, okay? Next year. My cousin Seth, Chris and John in Chapel Hill, my pickleball crew, some of them are here. John, Justin, Duran, Ian, Cal, CJ, and Emfrey, our neighbors, Mac, Justin, Will, Carrie, Raven, all my friends, cousins, and family in Michigan. All the speakers that spoke today to honor her. A huge hug and so much love to her lab that all became my friends. Jared, Taylor, Yang, Hannah Lee, Hyam, to her undergrads, Julia, Lauren, Dinah, Trinity, Victoria, Celia, Sydney, Logan, Melanie, Richard, Jasmine, Helen. I don't even know if I met all you. Everyone that helped set this up. And finally, to Natalia's family who are watching from Chile, I love you. I can't wait to see you, Matthias, in July, and Karina in December. And lastly, I'm just so proud of Natalia. We are all so proud of you. My dear Bolo, the cats and I will never stop loving you. Please keep finding ways to guide us, to visit us, to say hello. Thank you.
Y'all, can we show Trevor that our hearts are with him one more time, please? Thank you. And so, Natalia was a star. She was a star. And it is our hope that over the last few hours, that together we've been able to see her light. And that we've been able to feel her spark, to learn something new about her, maybe about mast cells, and to appreciate her legacy. A legacy that she built across all the phases of her life as a person and as a scientist and as a professor. How lucky we are. How fortunate to have had her as a part of our lives. And I give thanks for that. And how lucky we are and how fortunate it is that she was able to achieve her dream of being a scientist, to have her own lab. I just want to do science. And that is what she did. And I give thanks for that. And so, though the formal portion of our program is about to end, I hope that you'll stay for a while and enjoy the food in the back and the flowers that we brought. And the bar is going to be open, either in here or out there. We'll figure it out. And this reception will run until about 7 o'clock. And then afterwards, if you choose, there'll be the informal gathering at the Raleigh Brewing right around 8 o'clock. Okay, so we have opportunities to share your stories of her, right? And this is the day for it. Okay. Thank you again to all of our speakers. Thank you again to those individuals who made today possible. Thank you again to you for your presence and this time with us, both here in person and on live stream. And take good care. Thank you.
All right. Whenever you're ready.
I'm ready.
Let's roll.
Good times for a change. See the look I've had can make a good man turn bad. So please, 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 let me get what I want this time. Haven't had a dream in a long time. See the luck I've had can make a good man bad. So for once in my life, let me get what I want. Lord knows it will be the first time. Lord knows it will be the first time.
You nailed it, Lena.
Alright, I'm going to listen to it.
Let me stop listening.